For decades, the search for an effective Alzheimer's treatment felt, well, almost hopeless. It was just this cycle of failure.
Yeah, the clinical trial failure rate was absolutely staggering, wasn't it? Greater than 99%.
Just think about that number. Billions spent. Immense frustration for everyone involved.
Exactly. But finally, in the last couple of years, the story has really started to change.
It feels like we've hit this critical inflection point, and now, for the first time, we're actually talking about genuinely modifying the disease.
That's the monumental shift we're tracking today. Welcome to the deep dive.
Glad you're with us.
We've been digging through a stack of sources that chronicle this whole transition, from just managing symptoms,
Which it didn't do much.
right, to actively altering the course of the disease itself. And it's really being powered almost entirely by this revolution in diagnostics.
So our mission today is to unpack these breakthroughs. We're talking the first approved disease-modifying therapies, this blood test revolution with p-tau2, and the shift towards multi-target strategies. We really want to figure out what all this progress means for you, the patient, both right now, and, well, what this future brain health prescription might actually look like. Okay, maybe let's start by just quickly looking back at what our sources call the symptomatic plateau.
Right.
Before these recent approvals, the ceiling for treatment was it was incredibly low. What did that standard of care look like for all those decades?
Well, it was really defined by trying to boost whatever functionality was left, but it never actually stopped the underlying disease from progressing.
Okay.
The foundational treatments were these things called cholinesterase inhibitors. You might know the names: rivastigmine, galantamine.
Right, and the idea behind those.
It was based on this theory, the cholinergic hypothesis, that Alzheimer's involved a major shortage of acetylcholine.
That key neurotransmitter for memory.
Exactly. So the mechanism was pretty straightforward. Block the enzymes acetylcholinesterase that break down that acetylcholine, just trying to boost the signal that was still there.
Simple concept. But what was the real-world limitation? I mean, how much benefit did patients actually get from that?
Very little, unfortunately. It was modest. Then you also had memantine added later, usually for moderate to severe AD.
Okay.
And that addressed a sort of secondary problem, this excessive glutamatergic stimulation, basically excitotoxicity.
Frying the neurons, essentially.
Yeah, kind of. Memantine helped cool down that overstimulation a bit.
But even putting both approaches together, our sources still describe the benefit as just modest and temporary. It sounds like just delaying the inevitable.
Precisely. For the people who responded well, and not everyone did, they might get, say, 6 to 12 months of stabilization, maybe a slight pause in their cognitive decline.
But not a reversal, not stopping it.
No, absolutely not. The drugs did not alter the inexorable neurodegenerative course. It was this incredibly frustrating ceiling that really pushed researchers to take huge risks, looking for therapies that could actually, modify the disease biology.
Okay, so the limits of those old drugs were painfully clear. That frustration finally led us where?
To the paradigm shift we're seeing right now, the full FDA approval of the first anti-amyloid monoclonal antibodies.
These are the first official disease-modifying therapies, right?
That's the label, yes. And while they both target amyloid, which we know is a key hallmark of Alzheimer's, they actually approach the problem a bit differently.
How so?
You can think of it like dealing with debris, maybe. One targets the sort of sludge, and the other targets the more solid structure. Lecanemab, brand name lecanemab, it focuses on what are considered the highly neurotoxic soluble A-beta protofibrils.
Protofibrils, those are the smaller clump.
Yeah.
Before they form big plaques.
Exactly. Those small intermediate aggregates that float around causing trouble before they solidify into those dense plaques.
Right. And what kind of benefit did lecanemab actually show in its big trial?
It produced a statistically significant 27% slowing of clinical decline that was measured over 18 months in people with early-stage Alzheimer's.
7%. I mean, it's not a cure, but...
No, but it's substantial. It's the first time we'd really proven biological modification and a clinical benefit together. A landmark.
Okay. And then there's the other one, donanemab. That one has a fascinating twist in its approach, doesn't it?
It really does. Donanemab, brand name Kisunla, it targets the deposited plaque itself, specifically a form called pyroglutamated A-beta, found right in the core of the plaque.
So it goes after the established structure.
Yes, and this specificity seems to allow for very rapid clearance of that plaque.
Yeah.
But the really unique feature for patients is the finite dosing.
Meaning you don't take it forever.
Correct. The goal isn't lifelong treatment. Treatment is actually stopped once a certain level of plaque clearance is achieved on scans.
Wow.
Yeah, in the trials, nearly 3/4 of the participants were able to stop the infusion by 18 months.
That seems like a huge deal for reducing treatment burden and probably cost, too.
Absolutely. And donanemab's trial showed an even stronger effect, actually up to 35% slowing of decline.
But there's a catch, right? Only in certain patients.
That is the crucial caveat. Our sources call it the tau threshold. The benefit in the donanemab trial was pretty much lost in patients who already had high levels of tau pathology at the start.
Tau, the other misfolded protein in Alzheimer's of the tangle.
Exactly. This really reinforces the critical need for early intervention. You have to clear the amyloid before the downstream tau tangles really take hold and spread.
Because once tau gets too advanced,
The neurodegeneration seems to become self-propagating.
yeah.
And at that point, just targeting amyloid alone probably isn't enough.
Okay, so incredible progress, first real disease modification, but it comes with a significant risk we need to talk about. ARA, amyloid-related imaging abnormalities.
Yes, ARIA is the major safety concern across this whole class of anti-amyloid therapies. Shows up on MRI scans.
How does it look?
It's either ARIA-E, which is brain swelling or edema, or ARIA-H, which means microhemorrhages, little bleeds.
That sounds...
It can be. While it's often temporary and asymptomatic, detected only on the scans, ARIA can sometimes be serious, even rarely fatal. And the key risk factor we know about is genetics.
Specifically.
People who carry 2 copies of the APOE4 allele, the homozygous carriers, they have a markedly higher risk of experiencing these serious ARIA events.
Which means these treatments aren't simple pills. They require mandatory MRIs, baseline, and ongoing surveillance.
And really complex risk counseling. Patients need to understand the trade-offs. And we also have to quickly mention the controversial ghost in the room here, lecanemab. Aduhelm.
Right. That approval was...
It really was.
Yeah.
Lecanemab served as the field's most vital and, frankly, most expensive lesson in regulatory rigor. It got accelerated approval back in 2021 based purely on a surrogate endpoint.
Meaning it lowered amyloid plaques on scan.
Exactly. Its ability to reduce amyloid. But the clinical data was conflicting. The 2 big phase 3 trials had opposing results on whether it actually helped patients clinically.
So the approval was based on biology, not clear patient benefit.
Correct. And that controversy taught us a huge lesson. Biological change alone isn't enough. We absolutely must see clear, reproducible clinical benefit. The subsequent success of lecanemab, paired with all that controversy, ultimately led to aducanumab being pulled from the market.
So the lesson from aducanumab, biomarkers matter, but clinical results matter more. That discussion wouldn't even be happening without the real engine driving modern drug development, the biomarker revolution.
Absolutely.
We're now actually defining Alzheimer's by its biology using this ATN framework. Explain that.
Right. ATN stands for amyloid, tau, and neurodegeneration. It's a biological definition, not just based on symptoms anymore.
But historically, getting that biological proof meant difficult procedures.
It did. Invasive and expensive ones. CSF analysis via lumbar puncture, spinal tap, or peaky imaging scans for amyloid and tau. Powerful tools for research trials.
But not practical for widespread clinical use.
Exactly. Impractical for most people.
And this is where the breakthrough comes that really changes the whole equation. The democratization of diagnosis through a simple blood test. The marker everyone's talking about is plasma p-tau217.
It is genuinely transformative.
Yeah.
Study after study shows that plasma p-tau217 can distinguish Alzheimer's disease from other neurological conditions with incredibly high accuracy, up to 98%.
98% from a blood test.
Yes, a performance that's comparable to PT scans and CSF analysis. And here's maybe the most mind-blowing part. It can detect these pathological changes up to 2 decades before cognitive symptoms even show up.
20 years early. Wow, that accessibility led to that huge milestone in May 2025, right? The FDA approval of the first blood test to aid in AD diagnosis.
That's when it became real for clinicians, no longer theoretical.
Okay.
And the Alzheimer's Association moved fast. In July 2025, they released the first clinical practice guidelines for actually using these blood-based biomarkers, or BBMs, in specialty care settings.
So how do those guidelines suggest using them?
Essentially, they created a system. The highly accurate BBMs, like p-tau217, can be used as confirmatory tests. They tell the clinician, yes, this is Alzheimer's pathology. Then, maybe slightly less accurate or less specific tests can be used as triaging tools. They can help reliably rule out AD in someone with symptoms, potentially saving that patient the cost and hassle of a PT scan or spinal tap if it's unlikely to be Alzheimer's.
So biomarkers aren't just research tools anymore. They're becoming central to diagnosis and management.
They're becoming the central, accessible, organizing principle of the disease. It's a huge shift.
Okay, let's shift gears a bit then. We've established these anti-amyloid drugs are a powerful start, a real breakthrough, but they aren't the single cure-all, especially once you cross that tau threshold we talked about. So the field is smartly diversifying. looking at a truly multifactorial approach. If amyloid is being managed, what are the next big promising targets people are going after in the pipeline?
Well, since the clinical symptoms memory loss, confusion seemed to correlate more closely with the tau tangles than the amyloid plaques.
Right.
The biggest focus right now is on anti-tau therapeutics. Researchers are developing different approaches, things like antisense oligonucleotides or ASOs like BIIB.
How do those work?
They're designed to actually get inside the brain cells and reduce the synthesis of the tau protein itself, sort of turn down the tap. Others are more traditional monoclonal antibodies like donanemab that aim to maybe mop up tau outside the cells and prevent it from spreading cell to cell.
And we're also seeing work targeting neuroinflammation, right? The brain's immune system.
Definitely. The microglia, the brain's resident immune cells, they can be a double-edged sword, sometimes protective, sometimes damaging.
So the idea is to nudge them towards the helpful side.
Exactly. If we can optimize how microglia function, we might change the disease course. Researchers are targeting key microglial receptors like TREM2, using agonists to try and boost their protective cleanup functions.
Okay.
Another target is CD33. Antagonists are being developed to block its function, which might potentially ramp up the clearance of a beta as well.
And even within the anti-amyloid class itself, we're seeing some really cool innovation, specifically aimed at solving that ARIA safety problem. I'm thinking of trontinemab.
Yes, trontinemab from Roche. That's a perfect example of next-gen technology. It uses this innovative brain shuttle technology.
Brain shuttle? What's that?
They're essentially tricking the body's natural defenses, the blood-brain barrier, into actively transporting the antibody drug into the brain much more efficiently.
Okay, so more drug gets in.
Right. The hope is this allows for rapid plaque clearance, but with a dramatically reduced risk of ARIA compared to the first-generation antibodies like lecanemab.
That would be huge.
It would. And we're also seeing a big push towards more patient-friendly delivery methods, a shift to subcutaneous formulations.
Meaning simple injections instead of IV infusions.
Exactly. Like SE lecanemab and another one called remternetug,
Okay.
much easier for patients.
And we absolutely cannot forget maybe the biggest wild card in the whole Alzheimer's pipeline right now, the repurposed drug everyone's watching, semaglutide.
Yes, the GLP-1 receptor agonist, already famous for treating diabetes and obesity. Ozempic, Wegovy.
Right. Why the excitement for Alzheimer's?
The rationale is compelling. There's a very strong established link between metabolic dysfunction, things like insulin resistance, and Alzheimer's disease risk. Sometimes people even call AD type 3 diabetes.
Okay.
So the thinking is that semaglutide might help by reducing neuroinflammation, improving how the brain uses insulin, maybe other protective effects too.
And it's being seriously tested.
Oh, yes. It's currently in 2 large global phase 3 trials, specifically for Alzheimer's EVO-K and EVO plus grounds. The results from those are going to be absolutely huge for the field, one way or the other.
This next point feels genuinely revolutionary to me. The scientific community is now fully embracing the idea that drugs alone, even these new ones, probably aren't the whole answer. We're really moving towards a more holistic strategy based on solid data.
That's right. It's the non-pharmacological pillar coming into its own. Our sources really highlight the landmark U.S. Pointer trial.
Tell us about Pointer.
Its findings came out in July 2025, and they provided really robust statistical evidence that these multi-domain lifestyle interventions,
Like what specifically?
things like structured physical exercise, following the MIND diet, which is like a hybrid of Mediterranean and DASH diets, plus cognitive and social stimulation, and careful monitoring of cardiovascular health.
Okay, a whole package.
Yes, and the trial showed that this package produced a statistically significant cognitive benefit over 2 years in older adults who were at increased risk for cognitive decline.
So not just good advice anymore. Lifestyle intervention is now a scientifically proven foundational part of any brain health plan.
Absolutely, it's evidence-based. The ultimate strategy moving forward will almost certainly be combination therapy. Think about how we manage other complex conditions like cancer or HIV/AIDS.
Right, cocktails of drugs plus other support.
Exactly. This could mean combining, say, an anti-amyloid agent with an anti-tau agent. That's actually being tested right now in trials like the DIAN-2 Next-Gen platform trial.
Okay.
Or maybe even developing single compounds engineered to hit multiple targets at once. These are called multi-target directed ligands, or MTDLs.
So wrapping this all up, what does this mean for someone listening right now? Where do we stand?
I think it means we're finally leaving behind the era of just trying to manage the symptoms of what seemed like this single monolithic disease. The future is clearly heading towards a personalized brain health prescription.
Tailored to the individual.
Exactly. Combining that proven foundation of lifestyle modifications with specific disease-modifying therapies, drug based on your unique biomarker profile, which we can now check with that accessible p-tau217 blood test, and then also managing any symptoms as needed with the older drugs if necessary.
It's a huge leap.
It really is.
Yeah.
We move from just modestly slowing decline with the old cholinesterase inhibitors to achieving the first biological disease modification with lecanemab and donanemab, validated by this biomarker revolution. And now we're actively building towards a sophisticated, multifactorial future targeting tau, inflammation, maybe even metabolic factors with drugs like semaglutide.
But, and this feels like the crucial next question. With this democratization of diagnosis through blood tests comes a new, very immediate challenge. Our sources call it the democratization dilemma. The p-tau217 blood test promises to make diagnosis fast, cheap, and accessible, detecting the pathology potentially years or decades before symptoms. That's amazing.
Yeah.
However, the technology seems to be rapidly outpacing the actual readiness of our healthcare system.
That's the worry.
So here's the provocative thought for you to consider. If these new blood tests lead to a massive, sudden influx of early diagnoses, potential millions of people finding out they have the underlying pathology, how will our current healthcare system, especially primary care doctors, possibly handle that?
It's a huge question.
Think about the deluge of patients needing really complex risk counseling, needing genetic testing for APOE4, needing the specialized MRI monitoring required for the new drugs like lecanemab and donanemab.
The infrastructure might not be there yet.
Exactly. So what systemic changes do we need to start making right now to ensure that this incredible diagnostic breakthrough doesn't just create an overwhelming clinical bottleneck, a situation where millions know they had the disease early but simply can't access the necessary follow-up care or treatments? Something for you to mull over as you process this deep dive.